My dear students, welcome to this anti-ulcer series, and in part 2 today we are seeing H2 receptor antagonist. So the whole chapter is classified like H2 receptor blocker, proton pump inhibitor, anticholinergic and prostaglandin analogs. In your syllabus only H2 receptor blocker and proton pump inhibitors are there, and anticholinergic and prostaglandin analogs, they are not there. So in this chapter we are just seeing these two classes. The first is H2 receptor blocker or antagonist, I can say that, and the second class is proton pump inhibitor. And in today's segment, I can say that we'll be saying only H2 receptor antagonist. So I would like to remind you over here that how the blockage of H2 receptor, I can say that it is beneficial for us. So you have seen that histamine, which is released from the enterochromaffin cell. I discussed with you in the last lecture that histamine is released from the enterochromaffin cell, and this histamine, it binds with H2 receptor, and binding with H2 receptor activates adenyl cyclase that converts ATP into the cyclic AMP. Cyclic AMP further activates protein kinase, and this protein kinase activates the proton pump, and that results into the expulsion of H plus ion, and H plus further it combines with Cl minus, okay, and that result into the HCl. And in ulcer we don't expect more release of HCl because already erosions are there onto the on the surface of stomach, I can say that. Okay, just imagine this is a stomach, already erosions are there, ulcer is there on the surface of stomach, and that's why you want that there should be less release of HCl. And for that, one of the probable mechanism is to block the HCl secretion as blocking of H2 receptor. If you block H2 receptor, so whole process will block and there will not be H plus secretion. So you can consider this is a mode of action of H2 receptor antagonist. And now we are seeing the drugs which supposed to block this H2 receptor. So all the drugs they call as tridins, I can say that. So first drug is cimetidine, second is famotidine, third is ranitidine, and fourth one is a nizatidine. Actually, I'll first discuss with the basic pharmacophore, by which it is helpful for you to understand the whole structure, all the structure, I can say that. So you'll find heterocyclic ring. This linkage is common in all the drug, methyl, sulfur and ethyl linkage. Okay, you can see that this is common in all the structure, methyl sulfur ethyl, methyl sulfur ethyl linkage, I can say that. So the first common pharmacophoric feature of this class is that heterocyclic ring, then it is attached with the sulfur containing aliphatic chain, and at the terminal portion you will find a basic moiety. The basic moiety is somewhat extent similar to the guanidine. Okay, here you can see that it is somewhat extent similar to the guanidine. It is somewhat extent similar to the urea. It is also somewhat extent similar to the urea. Now I'll discuss the each and every structure. The first is cimetidine. So what it cimetidine, im for imidazole. So the heterocyclic ring, that's why is imidazole. Okay, cimetidine, and this linkage is common. Okay, so cimetidine, it gives idea that heterocyclic ring is imidazole. And you have to remember this terminal side chain, it is a cyanoguanidine. Actually in guanidine we expect NH over here. Okay, NH is expected over here, but it is not guanidine, it is somewhat extent similar to the guanidine because one carbon is extra over here. So somewhat extent this structure is similar to the guanidine, so I can say that cyanoguanidine is there. So in this way you have to remember the structure of cimetidine. Talking about famotidine, this famo is not giving any kind of a hint about the structure, but you have to remember this is a thiazole ring, and a thiazole ring contains the guanidine ring. This is a pure guanidine ring, you can say that. And at the terminal tail portion you'll find somewhat extent sulfonamidoguanidine. Actually NH is missing, so it is not completely guanidine ring, it is a sulfonamidoguanidine. Okay, Then talking about the next structure, ranitidine. In ranitidine, the heterocyclic ring is a furan, and the basic side chain which is present on furan is dimethyl amino group. And here you can see that urea type of the structure. Urea means what? NH, C double bond O, NH. It is called such kind of functional group is unit is called as urea. But here in urea, oxygen is missing. So I can say that this structure is somewhat similar to the urea, but not exactly urea. So in ranitidine, furan is heterocyclic ring, and terminal ring is nitromethylene urea. In nizatidine, okay, ni for okay, no relation icon again. Thiazole ring is there, similar to the ranitidine. Dimethyl amino group is here, and the terminal portion is again similar to the ranitidine. So difference between the ranitidine and nizatidine is of only heterocyclic ring. Here furan is there, and here you can see that thiazole ring is there. Okay. So in this way you can remember the structure. Cimetidine. First, if cimetidine, it gives idea imidazole. So except cimetidine, I can say that all this three structure, they contains basic side chain. Okay, this is a basic side chain. This is a basic side chain dimethylamino. The dimethylamino on the heterocyclic ring. Okay, so this way you have to remember the structure. Now we are talking about the SCR part. So you can see that all the four structure, you can divide that one like a heterocyclic ring. The middle is a flexible side chain, and the last is a terminal portion, or I can say that polar group. So we'll see the SCR of drug in this way. So first we'll see the SCR of heterocyclic ring. So what SCR of heterocyclic ring says that the first compound which was discovered was cimetidine. So SCR is saying that cimetidine contains the imidazole ring. So SCR is saying is that you can replace imidazole ring with the furan, okay, with the thiazole, and it gives potent compound. You can see that here imidazole is replaced with thiazole, imidazole is replaced with furan, and this such kind of replacement gives the potent compound. So this is the first point of SCR. Then the second point of SCR is that introduction of basic moiety. You can see that these are three compounds. These are three compounds where you can see that basic moiety is there. Basic moiety is there on the heterocyclic ring. So second part of SCR is saying that introduction of basic moiety on heterocyclic ring, it enhances activity. So these are the 2 points. The first, I'm repeating again, you can replace imidazole with the furan or thiazole ring. Second, presence of basic moiety like dimethylamine, it enhances activity. Now we'll be discussing SCR of flexible side chain. So I told you that flexible side chain which is common is this methyl, sulfur and ethylene linkage. So what SCR says that? SCR says that the chain can be, you can see that this is 1, 2, 3, 4 carbon long chain is there, okay. So chain could be 3 to 4 carbon, okay. It should not be more than that one. And you can see that in all the structure, the sulfur is not directly attached with the ring. It is 1 carbon away from the ring. So SCR says that the sulfur should be 1 carbon away from the heterocyclic ring. And the last point of SCR is that people, they try to replace sulfur with other isotopes like nitrogen, oxygen and carbon, but that result into the decrease in activity. It means that sulfur is essential. And at the end I can say that this part is essential because any kind of changes, it results into the decrease in activity. Now we are saying the SCR of polar group, or I can say terminal group. So this is a terminal group, or I can say that polar group. So you can see that in all the four structure, either guanidine is there or urea type of the structure is there. It means that presence of polar group or basic polar group, I can say that they enhances the activity. So this is all about the SAR of H2 receptor antagonist, and mode of action is that, of course, this drug, when histamine is released from enterochromafin cell, it binds with the H2 receptor and it activates the kinase, and kinase further activates the proton pump, and proton pump further pushes the H+ ion. So this compound, they compete with histamine and they block the H2 receptor. Blocking of H2 receptor then stops whole process, and there will not be release of H+, and H+ will not be combined with Cl-, and in this way there will not be the HCl. In this way, we are, by giving H2 receptor antagonist, we are minimizing release of HCl. So I hope that you understood this H2 receptor antagonist, and thank you for listening to this lecture.